If you want to understand cell potential, so we have to construct electrochemical cell. Example for the electrochemical cell is galvanic cell. We say galvanic cell. In the previous video, I was talking about two half cells. What are the two half cells? One is anode, other one is cathode containing containers. So now here is, I am drawing one half cell container where pure zinc plate is placed in zinc sulfate solution, aqueous solution with 1 molar concentration. In another container, in another half cell, an electrochemical cell is combination of two half cells, and here I am taking copper plate is placed in copper sulfate solution, aqueous solution, which is 1 molar solution. We say copper sulfate solution, aqueous solution, salt solution, and this is pure copper metal solid. Copper metal is a solid, and here is pure zinc metal is a solid immersed in the liquid. So solid liquid interface is happening. That means solid interact with the liquid. And this is one half cell taken in LHS, and this is another half cell taken in RHS. And both half cells are connected by using a salt bridge, which is inverted U-shaped tube, glass tube. We say this is glass tube, and this glass tube is inverted U shape, and it has both openings at the ends, and this glass tube containing a solution. So a solution of potassium nitrate or sodium nitrate, saturated solution of potassium nitrate and sodium nitrate or ammonium nitrate, saturated solutions. Concentration approximately 0.01 m, or you can start the concentration 0.1 m to 0.01 m concentration of the saturated solutions you should take. But how we can take these solutions in the tube that has two openings? So it is not possible. Hence, potassium nitrate saturated solution or sodium nitrate saturated solutions must be stuffed in agar gel or in gelatin, and these openings are closed by the cotton stuff. So why we have to use cotton stuff? The cotton stuff is permeable for the free movement of ions or cations, permeability we say. So here that means salt bridge interact with the solutions only in terms of ions, not salt bridge doesn't take part in the chemical reactions. The contents of the salt bridge do not react with the solutions. You must remember this. So then what is the function of the salt bridge here? Salt bridge maintain electrical neutrality of the cell. Main important point. So when does the salt bridge, and I'll tell you. So now here is two half cells are connected by a salt bridge containing a saturated potassium nitrate or sodium nitrate solutions. And the main function of the salt bridge is it maintain electrical neutrality of the cell, electrochemical cell. It maintains equilibrium between two half cells. And now this is one half cell, this is another half cells. Both are connected by. So now we have to join these two half cells. Two half cells we are joining by using conductor, and this conductor wire, copper wire, passing through a meter, and now it is connected to copper. Zinc is connected to copper through the wire through the ammeter to check the reading of the cell potential. So here the direction of, this is a negative charge and here is the positive charge. This is a cathode electrode, here is anode electrode. Anode electrode carries the sign of convention here negative and here is a positive. I was talking about in the previous video, zinc plate, at the center there is a negative charge and on the surface is a positive charge. The sign of convention of anode is a negative. The sign of convention of cathode is positive, but on the surface is negative. On the negative. Cathode positive, anode negative. Both are connected through the wire through ammeter. When two half cells are connected by the wire through ammeter, now circuit is made. Both half cells are connected, and between these two half cells there is cell potential. Cell potential has been generated. Cell potential has been generated. When the cell potential has been generated, so the electrons start moving through the wire from anode to cathode. Electrons start moving. Zinc loses electrons and copper gain electrons. So the electrons start flowing, electrons start moving from anode to cathode, where the charge will move opposite to the electron's direction. That means here charge flow opposite to the electron's movement. So the moment you connect the anode to the cathode, ammeter show the reading. The EMF of the electrochemical cell, EMF, electromotive force, or the cell potential is equal to how much? Around 1.10 volts. You can notice. The cell potential is 1.10 volts. So that means it generates current that is equivalent to 1.1 voltage. So how much this current? How the 1.10 volt? Let us look into individual cells or individual electrodes. Based on that, we'll calculate 1.10 of this galvanic cell. And here there are two electrodes are present. One is anode, other one is cathode. As you know that at anode always there is oxidation takes place. In one half cell there is oxidation. In another half cell there is reduction. That means, so at anode there is oxidation potential will generate. Oxidation potential has been generated at anode. At cathode, reduction potential has been generated. The sign of convention, how we show this oxidation potential? So electrode potential of zinc electrode potential, Zn get oxidation to form Zn plus 2, which is equivalent to plus 0.76 volts. This is oxidation potential of the zinc, which is taking place in one half cell. So electrode potential of zinc means oxidation potential of zinc, so plus 0.76 volts. Whereas in another half cell, what is happening here? Reduction is happening. So then reduction potential of copper, so copper plus 2 getting reduction to copper metal solid. This is aqua solution ions. So getting to this copper metal, which is equivalent to 0 plus 0.34 volts. This is a reduction potential. But as a sign of convention, we should consider either oxidation potential or reduction potentials. So now here is, as sign of convention, what we take, both half cells we should consider reduction potentials. So the reduction potential of zinc can be written as like this: Zn plus 2 Zn equal to minus 0.76 volts. And in case of copper, reduction potential is copper plus 2 copper metal solid aqua solution is equivalent to plus 0.34 volts. Now if you want to generate the cell potential, which is equivalent to how much? 1.10 cell potential, we have to write equation E cell is equivalent to difference of the reduction potential. Difference of the reduction potentials. So cathode reduction potential of anode reduction potential. Difference in anode reduction potential. So then E cell is equal to, we should write E Cu plus 2 Cu minus electrode potential of Zn plus 2 Zn. So how much? I told the values, single electrode potential values. Copper 0.34 minus 0.76 volts. So minus plus, how much you will be getting? So 1.10 volts, which is equivalent to E cell, which is 1.10, which is called cell potential. That means, what is meant by cell potential? Cell potential is the difference in the reduction potentials of anode and cathode. Difference in the reduction potentials of two half cells is said to be cell potential. Difference in reduction potentials of two half cells. Or we can define like this also: difference in two oxidation potentials of two half cells is equal to cell potential. Okay, so now we talk about standard cell potential. What is a standard cell potential? Standard cell potential. Standard cell potential. As I was talking about standard conditions, what are the standard conditions? 298 Kelvin temperature, 1 atm pressure, 1 molar concentration of solutions. If you measure the cell potential, that cell potential is equal to standard cell potential, which we represent E naught cell. E naught cell, which is standard cell potential. I think I made clear here. But now coming to this, so role of the salt bridge. What does the salt bridge will do? So when does the salt bridge come into the picture? So in one half cell, accumulation of cations increases. That means zinc plus 2 ions increases after progressing of the time. And in another half cell, there is deficiency of cupric ions increases. Cupric ions decreases. That means cupric ions number falls down in one half cell. Zinc plus 2 ions increases in other half cell. So when this happens, the cell potential doesn't work. Means cell potential ammeter doesn't show the reading. So in this case, salt bridge come into the play. What does salt bridge? And when the zinc plus 2 ions increases in this half cell, in order to balance, so nitrate ions pass into this solution through the salt bridge in order to neutralize. And where in case of cupric ions decreases, in order to balance cations in another half cell, so the potassium ions from the salt bridge will come into this another half cell. So potassium ions combine with sulfate ions, so neutralize. So that means electrical neutrality maintained through the salt bridge. Equilibrium can be established by using the salt bridge, and cell potential can be sustained for a long time because of the salt bridge. So here, cell potential, standard cell potential, most important thing. And if you want to calculate the cell potential, you must know the single electrode potentials values of the electrochemical cell.